Today is the last day of our 5-day long workshop on next-gen sequencing, big data, biology. And for last 4 days, we were listening on different methods and the advantages of next-gen sequencing and metagenomic analysis. Yesterday, we have learned about the advantages of NGS over Sanger sequencing and in various platforms like solid, ion torrent sequencing, nanopore sequencing, and so on. And so I think we all have got some idea on this NGS and metagenomic analysis. So this is the final day. We are expecting to learn more from Mr. Subhrajit Roy. And if we have any questions on past days discourse, the audience, the viewers are free to put those questions in our question answer box. So I'm handing over the mic to the resource person, Mr. Subhrajit Roy. So Mr. Roy, please start.
Thank you, Dr. Roy. So I'm sharing my screen. Good morning, all of you. So in the last 4 days, we have discussed about different aspects of metagenomics, where it is very important, why it is important to study, and where we can use this metagenomics. And it's a very young science in the field, and that's why we need to understand it more. And along with the advent of next gen sequencing technology, we have been able to explore it different aspects of metagenomics, and I hope it will gain some momentum in next few years, and it will become a more important a field of study. So today what I will be discussing is about metagenomic data analysis. So being a molecular biologist, this is not actually my cup of tea. So what I will be discussing today will be an overall introduction of how what kind of data we have and what approach we generally use for analyzing any NGS data or any data for metagenomic analysis. But the details of algorithm and the types of softwares we require for analyzing and what will be better, maybe the specialist on computation and biology and informatics can help you in a better way. So my idea or my intention is to guide you and let you know about what kind of analysis you should prefer, and what are the things you may face after you have performed your NGS sequencing. So as I have discussed in my previous talk, that microbiome is almost occupies in the entire Earth's surface and of course in the environment, and it is a different it is very essential part of our lifestyle, and even the presence and absence of any microbiome on us can have a huge impact on our health system, and of course it is important in agriculture and industry too. So the environmental DNA sequencing has revealed the expansive biodiversity of microorganism and clarified its relationship between the host-derived microbial community and host phenotype. Advances in the DNA sequencing and biocomputing enabled the exploration of the genetic diversity of the uncultured component of the host associated microbial community. As you know, the foremost important thing is that majority of the microbes cannot be cultured. So through the conventional genomic approach, we cannot go for get a comprehensive idea of what are the microbes are present in an environmental sample. So we need to have a high throughput sequencing data, or we can go for amplicon based sequencing. So the amplicon sequencing, for example, is the most widely used method which characterize the diversity of the microbiota. Say, for example, you are trying to survey what are the microbes are present in one site environmental sample, and having a taxonomical informative genetic marker like 16S rRNA, internal transcribed sequences, 18S rRNA. These can help you in a common to virtually, which are these markers should be virtually common to all the organisms which I want to study, or in other words, my organism of interest, and then target and amplify it by PCR and go for its sequencing. The resultant amplicons are sequenced and bioinformatically characterized to determine the microbes which are present in that sample and at what relative abundance. So both informations can be achieved by performing these kind of analysis. What are the components are there and at what relative concentration. Next way approach is the shotgun sequencing approach, which studying uncultured microbiota directly and avoids the limitation of amplification bias because it removes the step of PCR. So the DNA fragments that are generated and individual fragments that are sequenced, and using high throughput NGS technique, and finally each read is assembled to form an entire genetic profile. So these are the techniques which we generally imply. So the most primitive was the microbes when we go for culture. It is very high throughput and of course, but it's very expensive and laborious. And majority of the microbes cannot be cultured because we don't know the media conditions and right environmental conditions for its growth. So if we have the DNA as our starting genetic material, we can go for an amplicon-based sequencing approach where we can have a quick analysis, a survey of the entire microbial profile in a community, or through a PCR-based analysis, we can go identify the components. But the limitation is that PCR induces a bias. So you may not end up identifying the smaller fragments of smaller proportion of DNAs which are contributed by uh, the relatively low count of microbial population. So and the another approach is a metagenome approach, though it is very expensive and a little bit time consuming for to get an entire comprehensive idea. And we can go for a virome analysis if we are interested in viruses. And if we have an mRNA as a starting material, you can go for expression analysis or metatranscriptome analysis. And if the genetic material of the virus is RNA, then we can go for virome analysis too. So the three major questions generally we ask in a microbiome study is that who is there, what are they doing, and how they are doing. So I'm discussing this because you will find that in the next part of the metagenomic data analysis, the three aspects are very important. So depending on the question you ask, your analysis will be depending on that. So if you have a question, who is there, at what relative abundance of the community members are there, so you should take one approach. And if you have a question that what are they doing, to identify the functional basis of this, and how its functional chemistry is carried out, what are the environmental products that are consumed and excreted. So these kind of questions may be addressed through a different bioinformatic approach, and how they are doing it. So if you I need to identify a enzyme, a novel enzyme which is associated with any metabolic pathway, so these are done through a different approach. So this is briefly describing how metagenomic sequencing works, and what are the different workflows. So metagenomics sequencing ultimately gives you a huge amount of reads. So if you are present in my last talk, you will be able to know that different sequencing platforms can give about millions of reads, and from any environmental sample. So first you need to identify your good reads from those million of reads. So you need to have a quality control. Of course, it is not possible manually to identify which particular read is good, which particular read is bad. So what the thing is, practically we do, every time we get a read from an NGS system, it is the way the reads come out, it is assigned with a quality score. So if we have a software that can identify the quality reads for our next phase analysis, and thereby you can identify the poor reads from your selected group, and then remove the junk material and go for further analysis. Once you have the good read, you can choose who is there and what they are doing. If your question is to survey, to know, only to know what are the elements present in an environmental sample, so you can go for marker analysis, binning, and then genome assembly. If you have a question like gene prediction, then you can functionally annotate those sequences to a particular gene with a reference database, or identify a novel genetic signature. And ultimately, you can combine these two and go for a comparative metagenomics. So if you have all the information, say for example, you know what are the bacteria are present in one environmental sample, and you know what are the how they express themselves, what are the metabolic pathways they are associated with, and then compare two environmental sites or to different conditions to know what are the comparative metagenomics approach will be. And it might lead to metadata correlation or biomarker detections. Say for example, you have a patient affected with diabetes, so and you have a healthy control, so you collect their stool samples and then go for a metagenomic analysis. So the presence and absence of any particular microbiome in one of these two samples might help to determine which will be a good biomarker for diabetes, or it could be a prognostic marker that this particular person affected with diabetes will deteriorate more quickly than the other individual. So you can have a biomarker detection using this comparative metagenomic survey. So though despite of the fact that it is extremely beneficial to us, the metagenomics of course comes with a lot of difficulties and challenges. So the one of the most important challenges are there, like metagenomics data is relatively complex. So and large complicating data, and it's leads to an complicate the informatic analysis. For example, it can be difficult to determine the genome from which the read has been derived. The second is the diverse community type. The most communities are so diverse that most genomes are not completely represented by reads. So if you have done, if you have an idea or seen any metagenomic data, you will mostly find that the genome that has been assembled with the read counts are left incomplete. There will be some gaps which cannot be identified. So these regions or these unidentified regions might create a problem for metagenomic analysis. The third important challenge is the genome assembly. So when the reads overlap, so it is not evident that if they are from the same or distinct genome, which can challenge the genome assembly. So if you will find that when you extract the genetic material from one environmental site and ultimately go for sequencing, so multiple reads will be generated. So these reads might have overlap. So once you randomly fragment the genetic material, so you will have overlapping DNA fragments. So you will not completely don't have any idea once you assemble it using computation and analysis. Your computation part will be based on the reference genes available. So even after performing wonderful job by doing a perfect analysis, you will not be 100% sure that your genome which you have assembled is from one particular organism, or it is contributed, there is a contribution from other genetic material in it, though the proportion is relatively small. So whatever the information you will be getting is enough for your analysis, but still there will be some glitch left which will create a challenge in genome assembly. The fourth important part is that large input data requirements. So you cannot just start with a single sample. You have to go for a larger volume of data input to identify a meaningful result of it, because vast amount of genomic informations are being gathered. So to make an assembly of the genetic structure of making a genetic map, you need to have an information, huge amount of information. Otherwise you will be getting a fragmented, only a small pieces of a region will be there and it will be of no use. So the next one will be unwanted host DNA. So I have explained in my previous talk that how the presence of host DNA, if you are isolating samples from anatomical locations of humans, then it will be preoccupied by different and sometimes overwhelmed by the amount of host DNA material. So while doing a community analysis, you will find very hard to assemble or get a complete idea of the genetic data present in that sample. And the final is the metagenomic contaminants, which can inhibit your these elements are different elements or things which you don't want for your survey, and this will lead to misleading analysis of your community genetic diversity. So all these challenges, if you have understood, you will find that this is very hard for a biologist or a molecular biologist or microbiologist to do it all alone, because these data are itself very huge. And that's why we require a computational biologist, a bioinformatician. Well, it's welcome that if you are able to or capable to learn bioinformatics, and I of course appreciate if the students have any predisposition to bioinformatic analysis, learning languages that can help to analyze the data of their own. Of course, it is good. But in most of the cases, the biologists take the help of computational biologist or bioinformatician for analyzing their data, and it is very important to have a proper collaboration for that. So as I have told you that the data you get from your NGS data is huge. Even that a computer cannot store it. It generally store it in a cloud. So each time when you get your data, and if you have to assemble your reads, which is hardly 250 or 200 bases long, and then ultimately assemble into a complex community genetics, it will be extremely difficult. And it's like a jigsaw puzzle. You have to assemble one by one, depending how your contigs overlap, how your reads are assembled, and then trimming your adapters and indices from your reads, then going for an assembly, then going for a quality check, and functionally annotating it. So it's very overwhelming. So this is the general workflow what we do, and thanks to the bioinformatic tools available, which do this stuff for us, and you need a proper computational uh, a lab for that. And as you have, if you are going for an amplicon based analysis, you will have a paired read, and reads about 250 or 300 bases. So once you remove the barcodes, primers, and go for a quality control, there are several softwares available like KIME and usearch, and ultimately you can go for merged clean amplicons. And then either you can go for an amplicon sequence variant to know the what are the variants are present in the variable region of these 16s rRNA amplicons, or you can go for an operational taxonomic unit. So these two help you to assemble the phylogenetic profiling of your sample. So as you know, that amplicon based approach are essential for profiling what are the elements are present, what are the components are present in one environmental sample, and then you identify the genus or phyla of microbes which are present in that sample. So the next approach is the metagenome, where you have a shorter read, and still you have to go for a quality control and removal of the host genetic material. So and which is done with a different software like Bowtie 2 and Trimomatic, and there are other softwares too. So once you have a clean read, you can go for a curated database search, or identifying a novel gene from your config. So if you have assembly of your genome, you can help take the help of and advantage of the different databases which have the informations and functionally annotating your genetic features. And either you can go for a taxonomic profiling or identifying the functional pathway and go for a pathway analysis. And same for the contigs prediction using proca. Proca is for prokaryotes, and you can identify the contigs and go for a functional analysis. So I have several times I have mentioned in my talk that contigs. So what are contigs actually? So say for example, a chromosome is a very long molecule. Even if you are doing with microbes, it's still very long for a sequencing machine. So the chromosome is fragmented into smaller pieces, and the smaller pieces are easier to sequences. So once you have the sequence ready, so you reconstruct the original chromosome from those shorter reads. A contig is that physical map which results from putting several little overlap bits of DNA into a longer sequence. So this contig is a physical map resulting taking a small piece of DNA that overlap and putting them into a longer sequences. So as you can see here, how we assemble in those shorter reads into a longer genetic reads. I will give you analogy on that in my later slide. So these are the two approach when what we generally take. That is, one is amplicon sequencing, one is shotgun sequencing. If you have a microbial community and want to know only the operational taxonomic unit or only genus level specificity, then you can go for a 16S rRNA sequencing. But let me remind you, there will be significant amount of amplification bias present in it. So once you have your amplicons ready, you go for a uh, quality control and a cluster generation. Then you will be able to identify the features from it. From the shotgun sequencing method, same way you have to assemble your data and go for a quality check. Then using this, you can able to identify the species, strains, transcripts, functional modules, or metabolic pathway from that. So this is the 16S rRNA, and it has several conserved and variable regions. So generally, those who opt for 16S rRNA sequencing, they design the primers from these two regions, which has been shown in arrows. These are the primers from the conserved region, and they amplify these two regions and ultimately send it for sequencing. And there, the uh, analyzing the variants which are present in these variable regions will give you an idea of what relative abundance and what are the elements are of microbes are present in that sample. So either you can have your back, your microbes are maybe from this bacterial cluster or phyla, maybe from archaea, or maybe from eukaryota. So frequently when you will go through this metagenomic data analysis, you will uh, come through these terms frequently. So though I will describe it, but it's better to give you a brief uh, ex, uh, explanation on what are these elements actually mean. So the assembly means putting the sequence fragment of DNA together into a correct genomic position. So binning is the process of grouping reads and assigning them to a taxonomic unit. So you can group them according to their similarity, or you can go for a comprehensive grouping, or you can go for annotation. So once you have your genes assembled, once you have your operational taxonomic units ready, you can go for annotation. Annotation is the process of marking the genes and their biological features in the DNA sequences. So which one ADI is the coding sequence, which one will be the promoter, which one is the exons. These annotations are essential for once you have your data ready. So what is assembly? So as we know that we start our analysis with shorter DNA fragments or libraries, which are once has been read, are assembled into small contigs, and ultimately those contigs overlap and they are assembled into one functional genome. As you can see here, you have started with this black, which is represents the genome, and you have fragmented it into shorter reads. So from each of these reads, which has been generated through your next-gen sequencing technology, and ultimately through scaffolding, removal of the indices and adaptive sequences from your reads, quality control, whatever the things you've done, ultimately will be helping you to assemble your genome. So once you have assembled, you will be able to get an idea of what is the genetic material was actually present there. But still, as you can see, there is one small portion which was not able to assemble from your actual genetic material, though you have started with an intact genome. So this is because the read qualities in that regions will not be very good. So if every time when you do that, those regions will be kept as blank. So after repeated sequencing, you might have an idea of what are the things, but still, as in case of human genome, there are certain areas which cannot be sequenced. So still we are trying to identify those regions. So this is one of the drawback of metagenomic studies here. So if you are going for a 16S amplicon based data procuring, so either you can go for a closed reference study, or you can go for a de novo assembly. So if you are going for a closed reference study, if your 16S reads are represented by these red dots, and you found that your the genetic signatures which are which are represented from these red dots through your sequencing analysis are closely associated with this present material which is present in this sequence. So you have a closed reference on that. So you can use green gene or Silva for these softwares are available to make a closed reference sequencing from that. For the open reference sequencing, you can go for a de novo assembly. So you identify your genes which is not exactly matching to your reference data, and ultimately assemble with a de novo assembly cluster, and identifying a new genus or different species from that. So for the next part is a whole genome shotgun sequencing. So once you have your bacterial genome present in the samples, so you can fragment into smaller pieces, and ultimately these pieces are assembled into longer reads, and using computer assembled consensus sequence, you will be able to get an idea of what are the elements present in your sample. So as you can see, those circular DNA fragment bacterial genomes are present. And when you fragment and linearize into smaller DNA fragments and sequence them, so once the sequence data is available, you can go for a functional analysis and go for an assembly, and ultimately identify what are the elements present in that sample. So of course, depending on their similarity and clustering of how close the genetic material they are. So for the alignment, we must have a reference tool available. So these are the references which we use to align our sequence read with the available data. Like one of the most common we know so far is the BLAST, or basic local alignment search tool. Those who are accustomed with Sanger sequencing must have uh, idea how to align their genetic sequence read or chromatogram with the available genetic database. So there are, for the metagenetic studies, you have Bowtie, Diamond, and other available softwares that can help you to align your data. And your alignment should look like this somewhat, where it is represented by a hyphen, as you can see here. And if it is an exact match, it will look like this. And if you do not have an exact match, it will be left with some gaps. So the data which we get from our sequencing platform is like, as you can see, this is a FASTA format, which is presented by a greater than sign, along with an identifier, along with a species name, and which gene it represents. Then you have a text file of genetic sequence reads with no gaps in it. But the information or the data reads what you get from your NGS platform, particularly from Illumina, is a FASTQ format, which doesn't look like this. It looks somewhat like this. It has very short read, and the first line starts with this identifier. So this first identifier starts with an @ sign along with the sequence identifier. Next we have your so next you have your sequence present, and sometime it is present with a quality score. So depending on the quality scores are also known as Fred scores, which are basically printable ASC2 characters. So these are algorithms which are based on base calling error probabilities. And apart from that, if you are using nanopore, you will get an information as a FAST5 file. And these are different requirements for your data assembly. So if you have a whole metagenome sequencing or shotgun sequencing, so there are several tiers of your assembly or mapping your information. So if your information is based on a huge amount of data, it can be from a metagenome or metatranscriptome, depending on if you have initiated with rDNA or RNA. So as you can see, there could be a possibility of the presence of species 1, 2, and of course there will be some ambiguous read which doesn't correspond to any existing population, and of course there will be some unidentified genetic regions. So for the tier 1, where we want to know perform a taxonomic pre-screen, the reads are identified, and based on the specific markers, they are assembled into different community species. The tier 2 will be the pan-genome search, where we search the entire genome and identify that. So there could be a possibility of the different species. Note that in this case, you didn't have a species 2, which is missing from the F, from gene F is not species. So we don't get an idea of that. And from the tier 3, we have where we identify the align the class unclassified reads into comprehensive and non-redundant protein database. So this was I was talking about that assembly based, which is a de novo assembly. So for example, you don't have a reference available, or your sequence, whatever you have, doesn't correspond to any available reference database. So how should you, what approach you should take while doing this? Say for example, you have one sentence which has been fragmented in randomly. So you will your general approach will be assembling them and overlapping them until you get a proper a sequence which particularly makes sense. Say for example, you have a fragmentation of different of sentence like this. So either it can be assembled into, like here it has been shown, once upon a midnight, dreary, while I pondered, work and weary. So this is what is one possibility. Another possibility could be while I nodded, nearly napping. So this is also an assembly, but this assembly doesn't make any sense. So we will consider this as an ambiguity and consider this one which particularly makes sense. So we often use De Bruijn graph construction for your assembly, and this is an algorithm working by chopping the reads into short kmers to form these DBG, and finally inferring the sequences from this DBG. The term kmer typically refers to the possible substrings uh, in a computational genomics, refer to all possible sequences from the read obtained through DNA sequencing. So the De Bruijn graph can be constructed from longer or shorter reads. The first step is to select the kmer size and split the original sequences into its kmer components. There is available genome assembler present, which is SPADES or MetaSPADES, which is depending on this De Bruijn graph reconstruction. So once you have assembled your genome, you need to go for a quality assessment. So your quality assessment, there is a quality assessment tool available, which is known as QUAST. And this tool evaluates your genome assembly by various matrices. And if you have a good score, you can select that. Our next approach will be binning. So it is our attempt to assign metagenomic sequences into a taxonomic group. So it can be either performed by classified into taxonomic group or OTU, and either you can go for clustering into sequences that represents the taxonomic groups based on the shared characteristics. So first, depending on the methods used, binning may provide insight or the presence of novel genomes that are difficult to otherwise identify. Second, it provides insight to the distinct number of types of taxa in the community. While many approaches provide coarse resolution of taxonomy, some are capable of indicating strain level variations, though usually at the expense of fewer binned reads. Third, binning provides a way of reducing complexity of the data, such as the post-binning analysis can be performed independently on a set of binned reads rather than an entire population of data. So these are the two possible binning strategies which we imply. First one is clustering first, and one is assignment first. So one is taxonomy dependent, another was taxonomy independent. So so as you can see here, so this is based on, particularly based on similarity, how you have clustered, how this sequence have, if your different reads have a particular signature, you cluster them into a small group and thereby go for a taxonomic assignment. And for the assignment first, a taxonomy independent approach, what we do, we directly go for annotation and with the taxonomic assignment. So these are the tools which might help you to do your data sets and the shotgun sequencing with the taxonomic dependent and taxonomy independent binning. So there are softwares like Megan for alignment based, and tree filer, MATA, and for taxonomy dependent composition based things, you can go for Phylophyta. And for taxonomy independent, there are Tetra, SOMs, compost bean, abundance bean, metaclusters. These are also available. So once you have performed your binning, so you can go for genome annotation. So genome annotation or genome gain annotation is a process of identifying the location of the gene and all of the coding regions in a genome determining what these genes do. So once the genome is sequenced, it needs to be annotated to make a sense of it. And the genome annotation consists of three main steps. First of all, it identifies the portion of the genome that do not code for the protein. Second, elements identify the genome, the process called gene prediction. And third is the attaching the biological information to these elements. So we already have spoken about PROCA, which is a genome annotation tool, mostly for the prokaryotes. And your genome annotation can be of two different approaches can be there. One is structural annotation, one is functional annotations. So if you are having structural annotation, you assign the open reading frames and gene structures, coding regions, regulatory motifs. And for the functional annotation, you identify the biological functions, biochemical functions associated with it, and involved regulations. So the for functional annotation, and finally the pathway reconstruction, which is the ultimate goal if you're planning for identifying the pathway, if the gene, what pathway these genes are associated with. You can go for KEGG pathway analysis. There are several platforms, web-based platforms are available, which can help you to functionally annotate your data. So you can go for eggnog, trigger FAMs, and KEGG pathway analysis. So this helps you to ultimately functionally annotate your gene. So I will briefly describe, if you have your a 16S rRNA sequence data available. So what are the basic in bioinformatic flow through which you have to go through? Say, for example, first step will be validate the map for any error. You identify the errors in your file. And once it is there, you join the pair end reads, split the libraries into FASTA, pick up the taxonomic units, or OTU, or operational taxonomic units, represents the taxonomic group for each sample through binning, plotting the taxonomic summary, alpha rarefaction, and ultimately data submission to NCBI. So there is a sequence read archive in it where you can go and upload your data available until you have your publication ready. So this is how you do your bioinformatics studies. So thank you for listening patiently.
Thank you, Mr. Roy, for his this illuminating lecture. There are two questions. First question is, how can one overcome the limitations of mapping-based analysis?
Sorry, can you repeat the question for me again?
Sure. How can one overcome the limitations of mapping-based analysis?
Limitations of mapping-based analysis. So the only way of doing it will be repeated sampling until you get a complete idea of those regions. So the mapping based analysis will be mostly based on amplicons and short reads. So what will we have if you have your high GC rich content regions available? If you have your 16S rRNA genes regions available, those elements which will be extremely variable cannot be ascertained. So by repeated analysis and doing this assembly, you can reduce your the gaps which will be left.
Okay. Next question is there any range of acceptable quality score? It depends on what characteristics of the sequence.
Of course, there is a score, because if you remember, there are Fred score assigned to any particular Illumina sequence, and I cannot exactly tell you, because I haven't performed this, that what exact score should be there. That there is, of course, there is a score associated with, and we generally cluster the Fred score into uh, a, a plus 35, and if I am remembering it correctly, yes, no, so one will be plus 33 and one will be plus 64, depending on the Fred score available. So there is a algorithm available, which is represented by a probability error. And depending on that, if your read is poor, it can be, there is a software called Trim Galore, which can remove your sequence which is not good for your further analysis. They can remove it for you.
Next question is, what is the function of Bowtie software package?
Utility of which software?
Bowtie, Bowtie, Bowtie software package.
Well, I don't have any idea of this. Maybe Dr. Dash can comment on this.
Actually, Botai software has been used for the alignment of the sequence with the reference genome. So,
Okay. Oh, so no, the bow tie. Okay. I somehow. You can continue, Dr. Dash.
so actually, the top hat and bow tie, these software eventually actually align the reads with the reference genome
Hmm.
and make, and on that basis the assembly has been formed. So Botai software or Botai software and the higher version of Botai is a top hat. This software has been used for the alignment of the reads with the reference genome.
Okay, any more question?
Subrajit, one more
Yes.
one I want to explain the term OTU for our student because I think they heard the new term OTU, but I
So
think
yeah, OTU is actually the operational taxonomic unit. So this is a sequence which will be representing a particular phyla. And as you have seen that 16S rRNA sequencing is generally used for clustering. And with the variations, if they identify any sequence which represents or variation of sequence which represents a particular cluster is known as operational taxonomic unit.
that
Any question? Any more question? I have a novice question, Mr. Roy.
Sure.
Suppose I got two unique bacteria. Okay,
Yes.
just first time I am finding them. So there is no control sequence, nothing. So
Yes.
should I go for functional annotation?
No, before you go to function and annotation, you need to find out if there are available reference sequences available, and to identify, because even there will be significant difference in your, if it is even if it is completely novel, then at least some part of your genome will actually map with your the document which has been previously documented. So this will already because whatever new bacterial species are there, they have evolved from a
Right.
species. So some of the features will be already present there. So half of the work will be done if you have your reference gene available. So you go for an analysis. Once some of the regions will be left, then in only you can go for function analysis or annotations.
Okay, another beginner's question is that in assembly-based analysis, if no meaningful sequence is obtained, so should we repeat the entire process again?
You can go for trimming of the sequences any further by lowering the acceptable score, and then go for another assembly.
Okay.
So making it more stringent for the assembly.
Okay, I think there is no question, no more question. So if no questions are there, so I would like to invite Dr. Shabjit Dash to give the vote of thanks.
uh, yes, Subrajit, that is a very fine talk throughout all this, and I think will be very helpful for our students, for our to work on this metagenomics field. I think all the steps that you perform from DNA
Dr.
isolation.
Dash, Dr. Dash, I am just sorry, just I am interrupting you. One question is again coming. May I ask the question, Dr.
Sure.
Dash?
Yeah, yeah,
So,
sure.
if errors in DNA sequencing occurs, then OTU will be hampered or not?
Sorry, Dr. Ray, I didn't hear you properly. Can you repeat the
Oh,
question?
sure. If errors in DNA sequencing occurs, then OTU will be hampered or not?
The errors means, yes, if you were present in my last talk, you will be knowing that we actually what we do, we go for a massively parallel sequencing. So the error rate, what will be generated from an NGS data, will be less than 0.1%. Yes, of course, it can hamper your OTU, but because it depends on the variations which are present in it. But the chances of OTU hampered from your error from the reads will be very, very less.
Okay. No more question. So, Dr. Dash, please continue.
Okay, Shubhrajit. So we will now finishing our session, and it is a wonderful talk. I think we will meet again and for doing the experiments and for doing the computation analysis, and hopefully we will collaborate with you and the other groups to make a good research in this field. So many thanks to you. Thank you.
Okay. I'd like to thank you all and our MAKAUT webinar team for organizing such a nice workshop. I think the participants have learned a lot, and really, I have learned a lot, I must say. So, thank you, Mr. Shubhrajit Roy, again. And
Thank
so,
you. Thank you
we
so
are
much.
coming to the last part of our, here we are going to conclude the session.
Thank you.
Thank you,
Really,
ma'am. Yes, ma'am. Thank you all for participating in this 5-day workshop. So please give the feedback and the attendance sheet that we have provided the link, and we are processing the certificates. And once the certificate is done, we will be sending the links to for the certificates to each participants. So kindly wait for 1 to 2 days. We will process the certificates and we will give it to you. So give the feedback and please attend the attendance link for today. Thank you all for participating, and thank you, sir. Thank you, ma'am.
thank you, everyone. So,
So
Thank
we are finally closing, ma'am. Yes, ma'am.
right.
It is requested to all participants to kindly take note of the feedback link. We are closing the session. So thank you all for participating. Thank you.